I'm excited to hear now from our next speaker, who will be Diane Wong from Purdue University.
Lower this. Okay.
Do you want the lapel mic?
I think this will be fine. Okay, thanks very much to the NAPB organizers for the invitation to speak. I'm really excited to be here. This is my first time to this meeting, and it's been a real treat to see some familiar faces again, especially the friends and colleagues that I haven't seen in person since before the pandemic. Okay, so I joined Purdue in January of 2020, which, as you can imagine, was a delightful time to start a research program. And so my group, we collect data from focusing on crop genetics as well as physiology with the intention to develop new, hopefully simplified and parsimonious process-based models to try to understand the mechanisms that underlie crop G by E. But today, what I'll be sharing with you is not any of this. It's actually the most recent results from this side hobby project that I've had kicking around for a few years now that I keep coming back to. So the data that have been assembled for this project were originally funded from a grant that I wrote back in grad school with Susan McCooch, who was my advisor at Cornell. And our other collaborators on this project are Anna McClung and Jeremy Edwards, who are rice breeders down at Dale Bumpers National Rice Research Center. Most recently, we've had Sajjad Jamshidi, who is a postdoc in my group, join us, and this has really infused the team with new energy, and he has a lot of really good modeling skills to add. And just to give you a sense of how long this project has been around, Ranghui Han, who's pictured in the bottom right corner there, she most recently helped us out with some analyses earlier this year. And she was an undergraduate student originally on the project and helped us do some DNA isolation. She had since finished her bachelor's, took a year off, started and finished her PhD at Davis, and has joined BASF since then. So it's been a long journey, but I'm still somehow very excited about this. Walking around the poster sessions at this meeting, I didn't see a whole lot of rice posters, so I wanted to give you a little bit of background about rice production in the US. So rice is most commonly associated with Asian agriculture, of course, and the US accounts for only 2% of global production. However, we are an important contributor to the global market and are consistently in the top 5 rice exporting nations. Currently, we are number 5. We export primarily to Central America as well as Mexico, and the main competitors being the producers in South America. And so here, our study focuses on southern rice production, and this is the region shown in this figure. So this is primarily along the Mississippi River Delta as well as along the Gulf Coast. And in this region, producers grow varieties primarily of the tropical Japonica subpopulation. It's a fairly narrow gene pool. The breeding history of southern US rice is relatively short. Really, the efforts began in the early 1900s, even though rice production has been, rice has been grown in the US since the 1600s. And so, like I mentioned, the gene pool is fairly narrow. In 1990, a study showed that examined all the pedigrees of US rice varieties, traced them back to just 22 introductions from Asia. And so, I mean, it's been 35 or 34 years since then, so maybe there have been more introductions since then. But looking at this is the pedigree of all of southern US rice, and you can see there's not a whole lot of new material coming in, and this is chronological. And when we throw genetic markers on the germplasm, we find that genetic groups align primarily with market segments. So these are defined mostly by the grain length. So in the South, they mainly breed for medium and long grain rice. And there's not a lot of stratification associated with the breeding program, so the breeders are sharing germplasm. Okay, so this study really is motivated by this really interesting data set called the variety acreage reports. And so back in 2013, a colleague from AgriCon and I were kind of dinking around the internet, and we came upon these really interesting reports. And these are production level, these are market data. They're at county level, and they basically describe how much acreage was allotted to each variety that was planted at that county in that year. So this is an example here from 1983 in Arkansas, and you can't read this, but basically it shows you each column is a variety, each row is a county from that year, and the values here are the amount of acreage that was allotted to that variety. And so these were really interesting. We have no idea why people collected them, but we were very excited because they seemed pretty unique, and we thought we should try to get our hands on as many of these reports as we could. And so that began a long journey of sleuthing around and emailing loads of people in the rice breeding community, trying to figure out who collected these, who were the stewards of these data. And it eventually got me to Steve Linscomb, who was at the time the Louisiana rice breeder, a few years from retirement at that time. And so he very kindly sent me the contents of his entire file cabinet and said, maybe what you're looking for is in here. So I dug through it and found some really interesting stuff. But I did find, it was a treasure trove, and we managed to assemble these types of data going back to 1970, almost continuously. And so we wanted to do something with them. And so our objectives on the project were to, first of all, digitize and assemble these variety acreage reports. And as you can imagine, with that example shown in the previous slide, we had to do a lot of this manually. So me and a group of undergrads did this. And we wanted to genotype the varieties that were grown. That way we could have a sense of the genetics that were present at those counties. And then we wanted to develop models that integrated genetic variation, historic weather, and historic yields. Again, this is at production level with all the limitations that Charlie had mentioned before. And we wanted to use these models, if we felt okay about them, to address questions regarding the climate resilience of rice at the production level again. And so one example of a question is there a difference between older and newer varieties with respect to their predicted performance under future climates? And a couple of aspects of this system that gave us some level of confidence, or at least so that we felt okay going forward with this, is that rice has a very small genome compared to other cereal crops. You can see that on the top right-hand corner. It's inbred, which makes some things much easier. The breeding history is relatively short. It has a very tractable gene pool, especially when we're focusing just on southern US rice. And we managed to combine 4 decades of data across these 110 counties, which we felt gave us enough climatic variation to play with. And so putting these models together required us to kind of reimagine genetic variation. Again, this is county level, so we're considering these counties as sort of bags of alleles. So just to walk through a really quick and easy example, this is Clay County in Arkansas in 1983. And so for this county, they only grew 4 varieties, and they're up here: Star Bonnet, LaBonnet, LaBelle, and Mars. And the values here are the acreages that were found in that year. And so if we had a marker and we could code them binary, we could get then this weighted value for marker X. We repeat this for all county year observations across all markers, and we have this sort of historic county level allele frequency data set. And just to very quickly describe the overall summary of our production level data, we have historic yields from NASS, the National Ag Statistics Services, from 1970 to 2015 across our 110 counties. And then, of course, we have the combination of variety acreage reports, and then we genotyped the varieties. The combination gave us this county level allele frequency at about 5,500 SNPs. And then for weather, historic weather, we took from the PRISM database, and we focused on April and July mean temperature and cumulative precipitation aggregated at county level. There's a reason for these weather variables that I don't have time to get into. And in total, we had about 3,600 total county year observations from this historical data set. At the same time, our breeder collaborator Anna McClung gave us access to trial level data. These are from the Uniform Rice Regional Nursery Trials. These are also across 4 decades, and these are trials that are carried out at 4 states, 4 rice growing states in southern US rice growing region. And then we have GBS data on these as well, on 76 varieties that overlapped with our production level data set. And then for this, we have weather data from on-site weather stations. And from this, we have about 3,400 trial observations. Okay, so this is our general workflow. We trained a bunch of models, and again, these are not the usual types of models that we work with in my group. These are black box models, so no physiology or biology involved. For testing, we wanted to use our trial level data. This is a very different type of data set. These are not production level, these are trial level, so plot level data. I didn't have a lot of hope that this would work at all. So, but we're curious. And this occurred in 2 phases. So the first, we split them up into the popular varieties, meaning the varieties that were planted with a minimum % acreage in at least 1 county or observation, and then all varieties that had overlap between the 2 sets. And then we wanted to use this model to ask questions at the production level. So these are the 10 methods that Sajjad had come up with. And this kind of modeling is not in my wheelhouse, so I probably won't be able to answer questions about this. But our goal was not really to compare the models, but rather to create an ensemble model that we can use. So very briefly, these are the results from the training and cross-validation. These are the 5 models that fell under the regression-based model category. From my naive eye, pretty similar. So the top row is from the training, and the bottom row is from the cross-validation. These are the results from the decision tree-based models. Again, pretty similar, maybe some more differences, but the goal is to develop this ensemble model. So we felt fairly happy with the cross-validation performance, so we combined these into an ensemble model and used them for testing. And I'm just going to show the results right here. And I forgot to mention, all of the results I'm presenting today, my collaborators haven't even seen yet. We finished up this weekend. So you guys are the first to see. And so these are, again, the multi-state trials. And I really had very little hope that they would work out because the scales are so different. When you're talking about developing a model at the county level, this is super aggregated, right? These are actual producers that are growing these varieties, and so their managements probably differ. They may not have, well, I don't know. Well, I shouldn't say that, but anyway, the trial data, at least, they're highly managed, right? And these data are coming at the plot level, so there's a lot more. The spatial scale is very, very different, and I think in general, when you have a model and you're trying to make predictions that are at a more downscaled level, it's much harder than doing the opposite. So I just thought that correlation would be 0, and it turns out that it was okay. This is the result for all varieties, which is not as good, of course, so correlation here is 0.28. But when we look at just the popular varieties, these are the 22 popular varieties, the correlation is 0.81. So that was pretty cool, actually. So we used this to answer a question, which was, is there a difference between older varieties and newer varieties in their predicted performance under future predicted climatic conditions? And so for this, we created these year of release groups. So from 108 varieties total that I had scraped together year of release data like a couple years ago for fun, we have 5 groups here, group A through E. These are the ones that are released from earliest to latest. And so per group, we took the varieties in each group and we reconstituted these sort of synthetic county allele frequencies with equal contribution from each variety of the group. Because our models were developed at the production level, at the county level, it makes a lot more sense to have these, or at least in my mind, to reconstitute these counties versus trying to make statements about just a single variety, because that's like saying one county is growing only one variety. And then we use historic data and then future climate models to backcast and forecast the performance of these groups across the 110 counties of the rice-growing region under 2 SSPs. So these are socioeconomic scenarios. So imagine like optimistic versus pessimistic, so high greenhouse gas emissions versus much lower greenhouse gas emissions, and 3 GCMs, getting confused, GCMs, which are the global climate models. So these are the process-based models, the process-based climate models that are used to actually generate those predicted weather data. And just to provide a quick example of how to interpret the resulting plots, what you're seeing on the y-axis is the relative yield change, and the relative to what? Relative to the backcasted performance. So this pink line right here is showing the model predictions for using historic weather from the PRISM data set. And then you have this line right here, and then going forward for the forecasted predictions, you have two lines, and each of these represent a single SSP. So for the green, that's the more optimistic scenario, and then for the red, that's the more pessimistic scenario. And these are generated, or the weather data that's going into the model are from the 6th IPCC assessment, so CMIP 6, and they've been pre-processed and downscaled. And so here are the results that we just got over the weekend for the 5 groups. So we have group A and B up here. These are the earlier release groups, so A being the earliest, B being the second earliest, and then group C, D, and E are the later release groups. And you'll see that there appears to be a difference between the earlier release groups and the later release groups, with the later release groups showing an increase in the relative yield change as compared to the earlier release groups under future climatic scenarios. So, okay, so a couple of things to mention. These are, of course, compared to the baseline backcasted performance. So note that these varieties probably didn't exist back then, or maybe some of them did, but not all of them. And so I'm not sure that this baseline is the right one to use yet, so I would love to hear feedback about this. This is very much an ongoing effort. And so to draw some tentative conclusions, which I'm always kind of nervous to do, but there does appear to be a difference in this predicted performance between the older versus newer varieties, and the newer ones were the ones released in the last 30 or something years, so like kind of 1990 and on. And I haven't shared these yet with my rice breeder collaborators, and as soon as I get home from these meetings, I really want to kind of pick their brain and ask them, does this make sense, and what are maybe the potential reasons for this? There's still lots and lots to do, even though this has been kind of a long data journey. We're still very much working on this, including re-examining the weather data and asking, other variables we should consider, even though there was a rational sequence of sort of logic to come up with the ones that we did use. And then actually going back to what Charlie says, and this is very much my feeling as well, my sort of gripe with this kind of method is that it doesn't account for nonlinear responses of plants to the environment. And so as someone who really loves plant ecophysiology and kind of like feedback and feed forward across scales, maybe this is why we never finish this project, because I always find that that is my major limitation with these kinds of approaches. But I think we have to be cognizant of that, and so maybe thinking of ways to maybe not go all the way to 2100, or just applying some sort of biological sense to the way that we run these models and also interpret the results. But I think I keep coming back to these data because I'm very attracted to them in a way. I think part of that is because I like very much the idea that historic records or historic data can perhaps have their potential more fully realized with the augmentation of more modern data types, and that probably stems from some belief that you can learn something from the past. And so I think that falls very much in line with this session topic, which is predicting the unknown. And so I really like this comic. Oops, and it's gone. Where you have this little library and it says you can't learn from the future, but you can learn from the past, because we have lots of information from the past and really nothing for the future. So I'll leave you with those thoughts. Thank you very much. And my time has expired. Thanks very much for your attention.